So hopefully that's showing up for everyone. Perfect. Thank you very much for the invitation. I'm excited to be here and talk to you a bit about markerless motion capture and what I hope are some potential clinical applications for this research. And as you mentioned, I'm at Queen's University, and I do want to acknowledge that we're situated on traditional Anishinaabe and Haudenosaunee territory here at Queen's. So I think everyone here is quite familiar with human motion analysis and the potential that we have to use biomechanical data to inform clinical decision-making, but it's really not something that has been done widely to date. And I think that one of the problems with that is not that we can't do it, it's just that the technology that we've traditionally used, which has traditionally been marker-based motion capture, is very challenging to implement in a clinical setting. There are some issues around potential soft tissue artifacts and variability in marker placement that can make it challenging to pool data that's collected at different sites or by different users. It's also very time intensive, limited to a laboratory environment. And as you can see from this poor sample from our lab, it's not the most comfortable situation for a patient. And I think that that's an important consideration when we want to make this a clinically viable tool. So there are definitely multiple approaches to doing markerless motion capture. Inertial measurement units, I know is a big focus of many people's research. There's also depth sensors. And then what we're looking at is a type of markerless motion capture that uses video cameras. And we're specifically looking at this company, Markerless. They are based in Kingston. So full disclosure, it was founded by previous graduate students from our lab. So we're not involved in the development of the software, but we do have, a pretty close working relationship with the group, and they provide some in-kind support to the lab. So I'll give you a quick overview of how it works. It's a machine learning approach. So this system was trained on, I think they're approaching 1 million images. So still images from the internet, just photographs from the web. And they have an army of annotators who manually annotate key points on the body. And these key points there's about 120 right now, and they include internal structures, like an estimate of where the hip joint center is, as well as obviously surface points as well. And so those are just, instead of the markers, we now have an AI that's estimating where those key points are on the body, and we apply the same type of modeling and inverse kinematic approach that we would use with marker-based. It's just a different way of finding the markers. And I think that anyone who's been involved in marker-based motion capture knows some of the challenges in terms of palpating patients, putting markers on, markers being occluded, markers falling off. And when you move to a markerless approach, it really is just unbelievably easy and straightforward to use. And in case I haven't convinced you yet, this is a side-by-side of doing the same data collection, markerless and marker-based. You may have missed it. The markerless is already done. It took us 2 minutes to do 6 walking trials. The participant shows up wearing whatever it is that they're wearing, and we just start recording. We do a calibration daily, and we're using off-the-shelf video cameras. So right now we're using Sony RX02 cameras, but it's camera agnostic. So to get the same data, we could do it in 2 minutes as opposed to 45 minutes for a traditional marker-based data collection time. Okay, so it sounds really promising, but does it work? So we've done a number of validation studies to try to understand this. One of the first ones was comparing it to existing technology that's used to do clinical assessments right now. So we used a gait right mat, and we also looked at marker-based motion capture. And we found that the spatiotemporal parameters that we calculated with the markerless system compared to both of those alternative systems were extremely close. And where we see differences, we suspect for the gait right, for instance, that it's probably an issue with the gait right because the markerless and marker-based were quite highly in agreement. We also did a simultaneous markerless and marker-based study. So the same person, walking on a treadmill. This was 30 subjects, and we found that there were very few differences in estimating where the segments were. And also importantly, because this is using whatever clothing people are showing up in, we were interested to see what happens if we have someone come back over repeated times. So this was a smaller study that we did on 8 of us, and what we had the participants come back a week, 3 trials with a week in between, and we didn't tell them what to wear, and we found that the variability within a trial, so the step-to-step variability, was on par with the variability between sessions. And so this is really promising because it means that longitudinal data analysis and data collections are really feasible, and we're not worried about a different person putting the markers on differently and that affecting the overall results. And because clothing is such an important question around this, we did a more thorough investigation of the influence of clothing. So we had participants wear what we called sport and street clothing. So shorts and t-shirts would be their sport clothing, and then street clothing was whatever they showed up in that day. And again, we found that there was a minimal impact on the joint angles when the participants were wearing 2 different clothing conditions for their gait. So we're fairly convinced that it is giving us reasonable biomechanical data. And now the more exciting part is, what can we do with this? And I think one of the most exciting things is that we can get out of the lab. And this is not really something that we've done with motion capture very much to date. So this is our same setup. We take it with us. We can take it outside. There's no restrictions on lighting. It actually might even work better outside because the lighting is great. And this really allows us to change the kinds of data that we collect. This is what it looks like when we do a data collection out of the lab. So we have a little cart that you can see there. Everything fits into the cart. We wheel it across campus. It takes us about 20 minutes to set up. These are the Sony cameras. They're like little GoPro cameras. They connect to a switch, and then that can be controlled by a laptop. It's a very straightforward interface that's through Sony. And so you don't need a high-powered computer to control it. So all of the data is actually stored locally to the cameras. And so that allows us to be extremely portable. And we go back to the lab at the end of the day and download all our data and analyze it. And you might notice that people are holding up their phones to the cameras when they're starting. And this is something that we implemented to really facilitate this wide scale data collection. Oh, not letting me advance here. Here we go. And so one of the big challenges with this is now we're collecting up to 80 patients in a day. How do we manage that data? And so we're doing all of our consenting through REDCap on the phone. So the participants use their personal mobile device, or we have an iPad. They do their consent. It generates a QR code within REDCap, which is automatically detected in the video. And that allows us to link the subject demographics to the kinematics as well. So we've collected to date a large data set. This is really one of our objectives, is to build a large reference data set of a diverse patient or participant population. And we've been able to do this over 14 data collections. So we have over 450 subjects that we've collected in those 14 half days, essentially. So we're excited about the possibility for what we can do with that. Sorry, it seems to be advancing automatically, but one of the things we're interested in looking at now is what we can do in a clinical population. So most of our work to date has been in a control population, and there's certainly questions about how does this work for someone who has a different demographic, maybe a higher BMI than what we've tested in to date. So this was a pilot study that we did to see if we could measure biomechanics and joint angles in severe osteoarthritic population. So we had 30 participants come into our lab, and the way that we structured this was that the physiotherapists in the orthopedic assessment clinic introduced the study at the time of a clinic visit, and if they were interested, they just sent them straight to the lab. So they are not talking, we're not scheduled, there's no scheduling, we're just open 9 to 5. Whenever someone shows up, we will collect their data. It takes us about 30 minutes to do a data collection, and about 65% of that time is the consent form and the patient-reported outcome measures. So the actual active time of doing the data collection is about 12 minutes on average. So we had them do a number of different assessments: walking, a timed up and go, stairs, and a balance assessment. And as you can see, these participants are what we would typically expect for a knee replacement patient. So these are all people who have been referred to an orthopedic surgeon for a knee replacement, and they definitely have a more diverse BMI than we see in a typical university student population. The other thing that's great about the Markerless system, my video is lagging a little bit. I don't know if it is for you as well. Might just be the Zoom, is that there are no restrictions in terms of mobility aids. So it works with a walker, it works with a cane, and this was sort of the worst case scenario in our data set in terms of clothing, where this participant is wearing a skirt that comes below her knees, and it actually is still able to estimate where her joints are for that. So this is really promising for us that it does have clinical feasibility, that we're not asking patients to change, we just have them participate in whatever they show up with at the clinic that day. And so the big question is, how do the results look? How do the kinematic results look? So we used our larger data set that we collected from out of lab data collections. We selected an age match control group that was 30 participants, and we look at the individual waveforms for the OA group compared to a 1 standard deviation bar for the control group. And when we average them, what's very reassuring is that we're seeing the patterns of changes biomechanically in an OA population that we expect from our historical marker-based data. So on the left is markerless, on the right is marker-based from a publication from one of our PhD students, actually from Janie's lab, Janie Wilson's lab in Halifax. And we see the same trends where you have that reduced peak and you have that sort of stiff gait through stance. And so this is very reassuring that we're getting the same kind of clinical or biomechanical signals that we would expect with a marker-based system. The other thing that's sort of a nice advantage of the markerless system is that it really allows us to look at case studies. And so when you have markers and you've collected your markers and the patient has left, if you see a strange signal in your waveform, it's hard to know whether that was a problem with the marker, something that went wrong. With the video, you can really play it back simultaneously and see that these patterns that you're seeing are realistic. So you can see this participant at a self-selected walking speed had a very unusual stiff gait. But looking at the video, we could see that that is actually what's happening. When we asked them to walk faster, interestingly, that very stiff gait disappeared. And so this gives us, again, reassurance that when we're perturbing participants, we're actually seeing biomechanically what we're expecting to see. Yeah, and I just want to sort of highlight, you know, it allows us to do lots of other tasks. Occlusions are not really an issue. So having a chair in place, having stairs like this, we're not too concerned about losing markers when we're doing difficult or more challenging tasks. And part of this study was investigating what was feasible with the markerless system. Could we measure stair climbing? It appears that we can. And then also what is feasible from a patient population. So one of the things that we found is that this group really struggled with the stairs. And so it's a very challenging task for them to do. So it may not be as useful just because they're so far gone. I think it would be interesting to look at stairs for a moderate OA population, but really challenging for a severe OA population. Only 20% of our participants were able to go up and down the stairs cleanly. So some of the other things that we can do is we can look at a wide variety of tasks. And, you know, we have historically focused on knee osteoarthritis and looking at what happens after orthopedic surgery, like a knee replacement. But we're also working with Dr. Stephen Scott, who is a neuroscientist, and looking at stroke patients after a traumatic brain injury. And so that's where tasks like the mini BEST or the Berg balance scale are traditionally used clinically, but we can probably get a lot more data from having them perform something like a balance task when we're assessing them biomechanically. And we can also have them do a whole range of activities and assess that with the Markerless system. And so this is an example of how we've looked at the balance tasks. This was work from Kieran Everly, who's a master's student who's recently graduated, and he looked at static balance tasks and analyzed both the poses as well as the movement strategies, so the way people modified their position to maintain balance using principal component analysis, and looked at the main patterns of variation and how people achieved these poses and then maintain these poses. And so one of his findings, for instance, was that when we did a tandem stance, so standing with your heel to toe, the movement strategies that were used by male and female participants were significantly different. And so that's sort of the level of sensitivity of the results that we can get from the Markerless motion capture. Another interesting finding was that when he looked at quiet standing, one of the principal components that came out was how much your shoulders went up and your arms went out, which is actually breathing. So we were actually able to pick up breathing rate during the quiet standing task. So that's the level of sensitivity that we're seeing. And then, we're interested in doing a more quantitative assessment of these traditional assessment tasks. So something like a timed up and go. Right now, a clinician will just look at the total time in seconds or minutes of how long it took someone to complete this, but there's a lot of individual components of the timed up and go. And so using the Markerless approach, we can automatically detect the start and end of those subcomponents of the tug, as well as analyze a traditional outcome metric, which would be the total time that it takes to perform a tug. And so this, again, gives us a bit more data in terms of how we can analyze it, as well as standardizing it. So you're not relying on individuals using a stopwatch. And this is work from Megan Safich, who was also a master's student in our lab. And then I just want to highlight a few other things that we've been, it's kind of a fun technology. I'm not going to lie, we're playing around a little bit just to see what the limitations are. So this is Vajra Keller, one of our current master's students, and we were just looking at, can we analyze yoga? This is a very different kind of pose than you traditionally see with gait, and it's something that would be very hard to do with marker-based because you would be occluding so many markers during the very confined poses. And again, it looks like it's something that we can use for these different types of movements, these more complex movements, and it's very promising for quantifying other types of motion other than just gait. So I think that it's been quite promising in terms of what we think we can accomplish with it. And then you can actually look at the kinematics as well. So, we have a research question around, can we quantify a beginner versus an expert yoga participant? And so these are some of the things that we're exploring with this technology. And then, of course, there's always sports. So getting into the field, I think, is a really interesting application of Markerless technology. I think a lot about using this for in-game situations. So there's definitely limitations in terms of the volume that can be captured, but there's also ways around it with multiple cameras. And so this is something that sports teams are also interested in investigating because it's just so easy, straightforward. And I think it's also interesting to think about pediatric populations because I think they're very challenging. Sorry, this one is really laggy right now. And this is an example where we went out into the community during a science outreach event, and we did live data collections at the same time that we were sharing our research with the community. And so this is also, it's a great tool for engaging the public in our research, which I think is really interesting and important as well. It definitely makes pediatric research easier. It's quite challenging to put markers on a child. Anyone who's tried to do that, I think will appreciate it, but I will also acknowledge that the technology is not going to solve every issue with pediatric research. So when you ask a child to walk normally, you get varying interpretations of that. So it doesn't solve all problems. So I think one of our biggest challenges has been sort of shifting the mindset from marker-based data, and it was definitely a challenge to compare the markerless and marker-based directly in our first paper, because we don't really have a ground truth. We don't necessarily know that marker-based is correct. I think a lot of us accept a lot of it, but I'm sort of looking at markerless motion capture as a new paradigm, and we almost need to forget everything that we think we know about biomechanics using marker-based, because I don't want us to perpetuate any errors that are coming from marker-based. And so I think we definitely need to verify the markerless is making sense, but I think we almost need to start from scratch and compare the markerless data to the clinical outcome that we're interested in, and really start building those large data sets and the multi-center data that's going to allow us to sort of move motion capture as a tool into a clinical realm. I'm definitely going to acknowledge that anytime we're doing anything AI or machine learning based, we need to be really carefully thinking about sex, gender, ethnicity, skin tone, and making sure that we are not introducing any bias. I know that this is a huge concern with any medical algorithm that uses AI, is that you definitely risk hard coding any existing biases into the results. And so this is something we can't ignore, and we need to be really upfront about it. So we ask everyone to self-identify their biological sex, their gender, and their ethnicity when we collect our data. And again, Kingston is not the most diverse place, I will say. So I am very motivated to branch out into multi-center studies to increase our diversity. We're collecting videos of people. I think privacy is an important consideration. We're careful about how we store and manage our data. So we have REDCap for our clinical data, and then we have a hosted secure server that is able to host medical data. So that's the level of security that we're using for our videos. We also do have the participants now. The secret benefit of COVID is that everyone wears a mask. And so I think that's actually a benefit. It works fine with the mask, and it just eliminates some of that concern around recognizing individuals. Data management is not to be underestimated. These are big files, and so it's definitely a challenge. Just to have the server space for this. So it's something we always write into our grants, is server space allocation fees. And I just put change management down there because I think it's really important. Again, this is a new technology, and I've definitely worked with other centers that they want to replace marker-based, and so they just do a straight swap. So they put the video cameras where the marker cameras were, and it's not enough. It's a different type of analysis. You need to be closer to the participants. You're not worrying about the same types of occlusions. So it's just making sure that when we're doing this, we're thinking about it from scratch and not just a find replace for marker-based. Opportunities, I think, are vast. Obviously, I think you can all tell that I have drunk the Kool-Aid on this one. I think that the longitudinal data analysis piece is really interesting. It's something that we're starting now with our orthopedic patients, where we're getting them at the start of their wait list time when they're waiting for a knee replacement surgery, and we're planning to look at how and if they deteriorate while they're on the wait list, because right now wait lists are approaching 2 years in Kingston, as well as looking at changes postoperatively. And I think just being able to take this anywhere is so valuable. And the fact that the raw data is video means that as the algorithms improve, as we change things, if we identify a bias and we correct it, we can actually reprocess all of our data. And I've actually noticed a significant improvement in how well the tracking has worked with the Markerless system in the 2 years that I've been using it, because they are constantly adding new features. They're adding new annotations. So it went from 53 to about 120 features, and it really improved the way that the tracking is performed. Yeah, so I think our research goals in our lab at the Human Mobility Research Lab are really looking at this, building large data sets and multi-center research. So multi-center is something that we're really interested in. We have all of our participants consent to sharing data with other researchers. So it's part of, if they're not in this, if they don't want to do that, then they're not enrolling in our studies. We also have them separately consent to open access data use. So we could share the data online, have it available for all, for researchers anywhere in the world. And I think that this is, a really important and really exciting piece of it. And then where I think this is going, I mean, there's endless applications. Just one example, because I come from an orthopedic background, is the interest right now in developing patient-specific surgical approaches with surgical robotics. And I think that this is something that we need to think about when we're planning those surgical protocols. So right now they're planning a knee replacement surgery that is for a dynamic motion over the rest of your lifetime based on a static X-ray. And so we should probably be thinking about incorporating biomechanics into how we plan surgery and how we assess surgery to see if those surgical robotics approaches are actually benefiting the patients. So I hope I've given you some ideas about how it works and some potential clinical applications. My previous work in orthopedics has definitely made me very interested in the clinical piece of this. I know that we're all interested in the technology and the basic science, but what really gets me excited is when we can start to transition this to actually influence healthcare, which I think is really in line with what you as an organization are interested in. So thank you for your time. I really appreciate it. And please let me know if you have any questions.
Thanks a lot, Lisa. I must say it was utterly interesting, the presentation. It's the first time I have a complete page of questions. I need to limit myself and let the audience ask the questions first. But before getting into the question, just wanted to mention, like, by knowing of the type of research you're doing and also the concerns that you shared with us regarding considering diversity of basically the subjects and patients and everyone included in performing the research. Just wanted to say that as the TransMedTech Institute is focused on basically putting the medical technology or services in action and making them available to all the stakeholders of the healthcare system, they also have an expert in EDI that like helps the researchers within the ecosystem of TransMedTech to put into action basically diversity in research, to basically practice what they call nowadays in Canada GBA plus, gender-based analysis, into action and into forming the research. And that actually give us a good opportunity, and also the students and all the researchers included in this, to consider this very important factor of considering diversity amongst the subjects. So for that, I guess we can start the question period, and maybe the very first question might come from our co-host, Dr. Sobhani, if you can, and then we continue with the other participants of the event.
Sure. Thank you, Dr. Laende, for the very interesting presentation. My question, of course, I have, like Dr. Moibi, I have a couple of questions, but I limit to one, so I let the others to ask their question. But my first question is, can you speak about the pipeline of processing this data? For example, we train the deep network each time you collect data from new participants, and what kind of pre-processing you do on this data?
Yeah, so we are not involved at all in the algorithm. So the company Thea is completely independent, and we actually don't provide them any of our images, specifically because we don't want it to be trained to, preferentially work on our data set. We want it to be completely agnostic. So the training is done on an ongoing basis from them. So they're just using images that they get from the internet and annotating them individually. So very manual annotation, literally someone clicking, this is the left thumb on a few hundred images that day. So that's how the retraining works. It's completely separate from our application of it.
Sorry to interrupt. They use images to train, or they use videos?
They do not use videos. So it's all still images. So when the analysis is done, every frame is analyzed independently. So, and it actually doesn't use any information from a previous frame to influence how this next frame is. So that's an important point to bring up, is that I've had lots of questions about, would this work with an atypical movement pattern? It doesn't actually analyze movement, it analyzes pose. And so skeletal anomalies may be an issue, but it doesn't actually analyze movement. It analyzes pose in still images. So every frame from every camera is analyzed independently. And then the 3D reconstruction is performed to estimate that point in 3D space.
Okay, and what pre-processing you need to do before you pass the data to that software? No, nothing?
Just a calibration. So we do basically a daily calibration, just in case someone bumped the cameras overnight when we weren't around, or so we do a calibration. We use a chessboard calibration, so it's done automatically. That calibration file is applied to all of the trials that are relevant for it, and then we literally hit batch process, and it runs overnight.
Okay. Okay, thank you.
And then, yeah, the output are C3D files, which we then look at in Visual 3D if we're doing gate to do the event detection, or custom MATLAB programs if we're doing something like the tug analysis.
I see.
Yeah.
Okay, perfect. I leave the floor to the audience. I think Ali Rosbayani, he was the one who first raised hand. Is that? Okay, go ahead, Ali.
Thank you very much for the presentation. It was really amazing and exciting. I also have a lot of questions, but I'm going to limit them to one or two. But one of my questions is, have this type of approach been executed using image processing methods as opposed to learning methods? So this way, the training process could be bypassed and replaced with these real-time image processing algorithms, such as morphological algorithms or intensity-based transforms. Do you think this could be a better way to mitigate to move to more real-time situation? Because this way we will be able to bypass the training and reduce the computation load. Do you think this would be a good path for this type of approach?
Yeah, it certainly could be. You know, I'm really interested in the application. I'm not on the software development side, but that is certainly a limitation, is that it's not real time, unlike the marker-based. And it is something that could be moved towards. You know, one of the things we're investigating is how many cameras do we actually need? Could we get away with a single camera solution? In which case that would significantly speed up the processing. And I think it's always a trade-off, right, of cost versus the resolution of the signal that you're getting out at the other end. But yeah, I know that this is, and Thea is just the company that we're working with because they are literally down the street. But there are many companies that are attempting something similar to this and using different approaches.
Okay, thank you very much for that.
Yeah.
I'm just gonna ask, if it's okay, a really quick other one. Do you think this method is currently able to help with something like hand dexterity assessment or hand gesture recognition? Is it able to do that too?
Yes, so they are working on it right now. I believe it's in development, but they have shown me a prototype of basically being able to sort of measure grip and all of those independent. I think they're looking at every metatarsal separately. Yeah.
Thank you very much for your answers.
So the next question, I believe, is from Dr. Puya Amiri. Is it right?
Yeah, if you let me. Thanks, Elise. Really interesting. I hope there's a day that everybody starts using it and stop using the marker-based ones. Such a
Are you
pain.
convinced?
I am, but I have some questions first.
Yeah.
So two main questions that I have is, the first one is that how do you define joint angles here? Is it the same as that sequential Euler or Cardan that we have in marker-based? And
yeah.
then you showed us the first paper that you compared the markerless and marker-based ones. What is the range of errors that you see in those, especially in the angles that we are reliable on in the flexion in the sagittal plane? I guess in the other planes we can't really trust what we see, even in marker-based, right?
Yeah, this is one of the challenges, is that we see much more consistent patterns in the other planes in terms of the joint angles in the markerless compared to the marker-based. And it's sort of, you know, one of those things where we've accepted that marker-based is just highly variable in those. And so we're not fully convinced whether the patterns that we're seeing in markerless are truly more consistent in participants, or if it's just the way that the algorithm is working, that it's always, you know, it has a bias towards that pattern. So that's something to look at. But I mean, the range of errors is within the range of errors that we see with marker-based. And so I think that this is something where we need to build larger data sets to look at those out of plane angles and compare them to clinical questions to see if this is a true signal, or if those out of plane directions are so noisy that we need to sort of discount them the way we do with marker-based, right? So the joint angles are calculated essentially the same way. It's a model with the origin of the coordinate systems defined the same way that we do with marker-based. So we're able to match the approach markerless and marker-based when we compared it within Visual 3D. And so those are all things that are defined within Thea. So they've made the choices in terms of defining those origin points and the coordinate systems. Okay. So, I mean, if you wanted a different one, that is definitely something that you could talk to Thea about. You could do a transformation. But what it does do is it means that when you're doing multi-center studies, everyone's done it the same way. And so you've eliminated that, you know, slight discrepancy in the way that someone defined their coordinate system. Oh, they used this landmark instead of that landmark, which changes the angle slightly. And now, you know, we're comparing apples and oranges. So you're always, yeah, it's a compromise, right? Like we're giving up a little bit of control, but we're gaining consistency.
Do you have access to the raw data, the bony landmarks during the movement as part of that C3D file? So what does the C3D file gives you? It's the angles directly, or
it gives you the pose matrices of the segments. Okay. Okay.
And by pose matrices means, okay, but for each segment separately for its own based on the landmarks that they define.
Yeah.
I see. And how much is the resolution of the angle that these can measure? Especially in something like a standing, the movements are quite small. So like in the order of 1 or 2 degrees, what is the smallest angle that you can measure? Does it depend on the segment length, or?
Yeah, I'd have to go back and look at the original paper. I think, I mean, like we looked at it and it was basically better than what had been within the ranges that had been reported for marker-based. And that was sort of our basis for it.
Thank you. Do you mind sharing the paper title at the end so we can go take a look?
Yeah.
Thanks. Thank you.
Great. I see another question from Mariam Ashbus.
Yes, hello. Thanks. Hi, thanks, dear Alice. Your presentation on your project was really interesting to me. And during your presentation, I was thinking that how much it can expand the application of movement assessment, especially in, I think, sport biomechanics, because it was really limited so far because of the in-field assessment were not really available. And I think method can really open a new door for sport biomechanics. But the question I have is that have these data been used for, I mean, the collected data in your approach have been used to be imported in other softwares, for example, which are used for surgery simulation or diagnostic application, that really some other softwares are needed. Have this data been used so far for this application?
Sorry, imported into diagnostic software, you said?
Yes, yes.
The previous one.
Yes, yes. Sometimes we need to collect data with the method you proposed, but we need to import them in some other softwares for surgery simulation or some other diagnostic approach. How easily it can be done, or have it been done so far?
Yep. So the output from Thea are pose matrices. So it's essentially a text file of the 3D coordinates and orientation of each segment. So because the Thea software was developed sort of as a spin-off company from C-Motion, which makes Visual 3D, they set up their output files. So they're called C3D files. They're essentially text files that they work very easily with Visual 3D, which is a pretty standard package for reading in biomechanical data and then doing further processing. Within Thea, you can also look at, it also has a visualization tool, so you can look at joint angles and all of those things within that software as well. So in terms of exporting, like we export to Visual 3D, we export to MATLAB, we export to R. We're still doing sort of basic science analysis at this point. In terms of, so it's a fairly open data format. In terms of using it further, I think the challenge becomes taking the raw data and putting it into a format that a clinician could use. So if we're looking at the clinical tool, you're not going to give someone raw pose matrices and ask them to make a clinical decision based on that, obviously. So I think you bring up a really good point in terms of what's the next step in terms of using this data. And that's something that we're actively looking at. But I will just bring up one idea, which is that we've been talking to researchers in Calgary who study patients with cerebral palsy. And that is, I think, in my experience, one of the few applications where they really use gait analysis to inform clinical decisions. And so they use marker-based right now to look at patterns of gait. And so we've shown them some of our results. We've tested it on an individual with CP, and they're saying that they're picking up the same patterns in their kinematic analysis that they look at that they think would be usable in their clinical tool. But that's a very specialized case where you have clinicians who are trained to analyze gait waveform patterns, essentially. And I think we need a different kind of tool when we're talking about clinicians in general. And I think that's a very open question.
Yes, great. Thank you.
Thanks.
Okay, to respect the time schedule, I think we have time for one more brief question. I leave either Puya or Dr. Moebi, whichever. I leave it to you to decide which last question.
I lowered my hand. I will pass, you go.
Oh, thank you, Puya. That's very kind of you. Okay, so actually I had a lot of questions. Some of them are already answered with the questions of other participants. So in connection to the previous answer that you provided regarding using basically data on the CP subjects and everything. So from the presentation, from the videos and pictures that we saw from the presentation, I guess like all of the subjects, or maybe most of them, were healthy subjects in terms of human motion. They did not suffer any neuromuscular deficits or neurological deficits. So what happens if we have like deformation on the body of the patient, or we have some limbs lost, basically? Is that tried already or not?
We haven't looked at amputees specifically, but that is an open question. We did test it on children. It was never trained on images of children, and it worked on children with no issues. So that's promising. But again, it comes down to the modeling. And I agree that if you have significant skeletal anomalies, that is something that is an open question in terms of investigating how well it would work.
And one other thing is that I saw from the photos that there was a very nice skeletal system showing the movement and everything. Was there an actual training on basically medical images to link the skeletal system to the motion, or it was just a way of showing how the marker less
Yeah.
systematic. Yeah.
No, it's just a visualization tool. And I've had other people comment that it's a bit misleading because it implies that it knows the bony locations when it is trained on landmarks.
Okay.
So no, it has not been trained against medical imaging, which I think is an interesting question. And we do have a dynamic X-ray system that just got up and running next door. And so that is definitely on my to-do list, is to compare how well it performs compared to a ground truth like that. Yeah.
Perfect. Thank you very much for the answers. It rests with me a couple of other questions, but I'm going to abstain myself. Maybe
Well,
later on.
Feel
offline.
free to reach out anytime.
Thank you very much for your kindness.